epidemiology is to study disease and health states really, and the spread of the disease. And to that end, we talked about where we find disease, the reservoirs that disease are found in. We talked about how disease leaves those reservoirs, so transmission and modes of transmission, and then how disease gets into the body or the portal of entries. Introduced a lot of terms. Many of the terms you're probably familiar with, but be sure that you do understand those terms. Prevalence, incidence, epidemic, pandemic, outbreak. We discussed factors that influence the spread of the disease, types of studies that can be done, and the organizations that are involved in monitoring disease. Be sure that you're familiar with each of those organizations and how you would be able to identify. One of them is, of course, a national organization, the CDC. The other is a local organization, the Public Health Department. And then the third is a worldwide organization, WHO, Worldwide Health Organization. That brings us to the end of that chapter, a discussion on nosocomial infections. These are infections that happen in the hospital. These are hospital-acquired infections. And why do they happen there? Well, because one, you have a lot of susceptible people in a hospital. You have a high density of these sick individuals, and therefore then the pathogens are in the environment. They're on the fomites, they're in the air, they may get transmitted via the individuals in the hospital, the other patients, or the health workers. Many of the pathogens that make up these nosocomial infections are part of your own flora. When you think about the reservoirs that we find in the hospital that can lead to these kinds of infections, other patients play a major role in providing the reservoir. They're coming into the hospital, they're sick. But we have to recognize that the hospital environment itself can play a role as a reservoir. Even though they're continually cleaning certain surfaces and things like that, doesn't mean every surface gets cleaned. Pseudomonas aeruginosa is a microorganism that can survive in environments where there's very little water, very little moisture available. It can absorb stuff from the air. So one of those microbes that can resist certain environmental conditions and hang on there. Healthcare workers, great source of nosocomial infections. Failure to wash your hands between patients, moving from one room to another, you can transmit microbes into those rooms. And as I mentioned, the patient's own flora. Some of the things to consider when we talk about transmission. Of course, the healthcare worker, the patient, can serve to transmit it via direct contact. But medical devices can also serve as a means of transmission. Catheters, IVs, all of these can have microbes on them. Respirators. And we have to remember then also that there are microbes that are going to be airborne, and that is another means of transmission. I have a little video for you I'd like you to watch. Again, it's a YouTube video. It's on nosocomial diseases. It's only about 6 minutes, won't take us too much time.
University Health Organization Management course 5308 presents nosocomial infections. What are they, who's at risk, and how do you prevent them? This presentation was created by Ron Chai, Brittany Chan, and Laura N. Leva. For sources, please see the works cited page included with this presentation. Imagine going in the hospital because you contracted an illness. You're admitted, and after a lengthy stay, you're dismissed, only to return and find that you contracted a nosocomial infection. A what, you ask? A nosocomial infection. What kind of infection is that, and how did I contract it, you ask yourself? We're going to cover this topic along with risk factors, consequences, and how to prevent these types of infections. Nosocomial infections are defined as infections that occur 48 hours after initial hospital admission or within 30 days of coverage. They're also known as hospital-acquired or hospital-associated infections. Patients that are at highest risk for nosocomial infections are the immune-compromised, the very elderly, and the very young. So how did I contract this infection? Did someone else pass it on? Did I touch something that was infected? You discovered that there are 4 main routes of transmission. These routes are contact, vehicle, airborne, and vector-borne. Contact transmission is the most important and frequent means of transmission and can be divided into 3 subgroups, which include direct contact, indirect contact, and droplet contact. Direct contact involves direct physical transfer between a susceptible host and someone who is infected. For example, direct contact occurs when hospital personnel are turning patients, giving baths, changing dressings, or performing other procedures. Direct contact is not limited between personnel and patient, but can also occur between patients. Indirect contact involves personal contact of the host with contaminated objects, such as instruments, dressing, or other infected materials. If proper care is not taken, hospital personnel can contaminate objects when assembling or handling critical equipment, such as respiratory therapy equipment or cardiac bypass pumps. Droplet contact is when infectious agents come in contact with the conjunctive eye, nose, or mouth of a susceptible host as a result of coughing, sneezing, or talking by an infected person. Droplet contact is considered contact transmission since droplets usually do not travel more than 3 feet, and close contact usually means within 3 feet. The other 3 main routes of transmission are vehicle, airborne, and vector-borne. The vehicle route applies when disease is transmitted through contaminated items such as blood. Airborne transmission occurs by dissemination of either droplet nuclei or dust particles in the air which are inhaled by or deposited on the susceptible host. And vector-borne transmission uses disease-causing organisms that carry the pathogen from one host to another. Many factors promote hospital-acquired infections in healthcare settings, including decreased immunity among patients, as well as diagnostic and therapeutic procedures, invasive techniques, and medical devices that increase the risk of infections. For example, patients are at increased risk of infection from invasive devices such as intravenous needles that are inserted through a person's skin, bypassing the body's first line of defense. The 4 major types of hospital-acquired infections are related to invasive or surgical procedures and include surgical site infection, central line-associated bloodstream infection, catheter-associated urinary tract infection, and ventilator-associated pneumonia. This pie chart shows the most common infections reported as acquired in a hospital setting. Some of the implications of nosocomial infections include cost, death, morbidity, and imposing community risk. Nosocomial infections are costly, with the direct cost of hospital-acquired infections in the U.S. estimated to be between $4.5 billion to $5.7 billion per year, with an average cost of infection of $13,973. The CDC estimates that over 2 million nosocomial infections occur yearly, leading to 99,000 deaths per year in the United States. Nosocomial infections also cause mortality. The cause-fatality rate for patients with ventilator-associated pneumonia is 42%. For nosocomial bloodstreams, the case-fatality rate is 14%. Nosocomial infections contribute to increased morbidity, including delayed wound healing, delayed rehabilitation, increased exposure to antimicrobial therapy, and its potential adverse effects and prolonged hospitalization. The average prolongation of stay is 3.8 days for urinary infection, 7.4 days for surgical site infection, 5.9 days for pneumonia, 7 to 24 days for primary bloodstream infections, and increased ICU stays of 8 days. Nosocomial infections contribute to the emergence and dissemination of antimicrobial resistant organisms. Antimicrobial use for treatment or prevention of infection facilitates the emergence of resistant organisms. There are many ways to prevent the spread of nosocomial infections. One method is to use isolation precautions. There are 3 kinds of isolation precautions: airborne precautions used for agents that can be transmitted through suspension in the air, droplet precautions used for disease spread through respiratory secretions, and contact isolation used for pathogens spread by direct contact. Hand hygiene is also very important. When hands are visibly soiled, the CDC recommends washing your hands by wetting applying soap and rubbing your hands together vigorously for 15 seconds, making sure to cover all the surface area on the hands and fingers. Rinse hands and dry them with a disposable towel, using the towel to turn off the faucet. Hands should be decontaminated before and after patient contact. The use of gloves in the hospital is also important. Gloves should be worn when touching body fluids, blood, broken skin, or mucous membranes, and should be changed between procedures on the same patient, after contact with contaminated surface, and of course between patients. Gloves do not replace hand washing, thus hands should be washed after disposing of gloves. Another method of prevention is environmental or surface sanitation. The CDC recommends use of EPA-registered hospital disinfectants to clean non-critical medical equipment surfaces. Surfaces such as walls, floors, and tabletops should be cleaned on a regular basis, while cleaning highly touched areas much more frequently. Appropriate handling of soiled linen is a part of hospital standard procedure. Linens including bed sheets, blankets, towels, and scrubs may harbor large numbers of infectious agents and must be dealt with properly. Soiled linen should be handled as little as possible and should always be kept physically separate from clean linen. Linen should be washed in water at least 160 degrees Fahrenheit for a minimum of 25 minutes. The first step in stopping the spreading of nosocomial infections is to have an understanding of what nosocomial infections are and how they are transmitted. By understanding how these infections are transmitted, you can be better prepared to stop the spread of these infections. Be sure to do your part to prevent the spread of nosocomial infections by taking preventative and precautionary measures.
She was in a little bit of a hurry. Yeah, she did talk a little bit fast there, but I felt it was a good review of the material that we covered last time. And one thing to keep in mind, these practices don't only apply to the hospital, you can employ them at home as well. Any questions, any comments? Okay. So that brings us to our next chapter, antimicrobials. So we're going to talk about agents now that inhibit or kill bacterial growth. Paul Ehrlich in 1910 wondered, knowing that if you use different stains, cells would stain differently. Recognized that meant that there had to be structural differences in the cells. Wondered then, if there are structural differences in the cells, could we find an agent, could we find a chemical that would selectively kill cells? Maybe kill one type of cell, but not another type of cell. He was the first one to introduce this notion of using antimicrobials. In 1910, used a compound called arsphenamide. It was a sulfur product that was effective in killing syphilis. So it was used against syphilis. And then a little bit later, Alexander Fleming was observing the growth of penicillin on a petri dish and noticed that the bacteria that were growing on the plate were not growing around the penicillin. There was an area where the cells were dissolving. And so he was the first really to discover this notion of an antibiotic. An antibiotic, a compound that is being produced by an organism. So an antimicrobial is an antibiotic, but not all antibiotics are antimicrobials. In this case, we're talking about something that is being produced by an organism. An antimicrobial is that and/or chemicals that we might use to inhibit or kill bacterial growth. So penicillin was the first antibiotic produced. It's been effective against many bacteria. Streptomycin came along later, a soil bacterium, or a product of a soil microbe. In fact, most of our antibiotics come from soil bacteria. We can take these microbes, we can isolate these selected microbes, and we can grow them in large vats. And from these large vats then, we can extract the antibiotic that they're producing. And sometimes we can modify that antibiotic so that it